All right, good morning. It is Monday, April 5th, and we are on to the fourth section of this class where we are going to do the respiratory system and urinary system. If you've looked at the schedule, you can see that there are bunches of assignments that we are going to be doing for this. Both systems have two unit reviews that you're going to be doing. Both systems have uh, 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 lab exercises you're going to be doing, PhysioX, a Labster activity, two PhysioXs actually, and a Labster activity, and a homebrewed version of a lab that hopefully you'll definitely be able to do some of the activities of, and hopefully all of them. So we'll talk about that in just a second. So again, we're going to start with the respiratory system, talking primarily about anatomy of that today, and then we'll get on to physiology and control over the next two lectures. Then we'll move on to the urinary system. On Wednesday, your first unit review is going to be due. On Monday, 1 week from today, your second unit review, and there's a control of respiration lab. Now, there's some caveats I want to give you with this control of respiration lab. First, do not do it unsupervised. Make sure there's at least one other person around when you're doing these activities. If you have any type of respiratory issues, if you have asthma, COPD, if you have a cold that you're getting over, please do not do these activities. Be seated while you're doing the breathing activities. One of the activities is asking you to hyperventilate yourself. That's why you want to make sure you have a sitting when you're doing that and have someone observing you, because there is a small chance that if you do it too much, you can actually cause yourself to pass out. The goal is not to get yourself to pass out, but to hyperventilate. Sorry? Okay. So make sure you're doing that carefully for the first activity that you'll be.
Some at the Jet Center.
Hold on.
They have coffee.
Okay, I'm guessing those aren't questions, so let's go ahead. I'm going to go ahead and mute everybody for right now, and then if you can, always feel free to unmute yourself if you have any questions. All right. So that is the first activity where you're going to be holding your breath and doing those. You will also be exercising and doing some activities. Also make sure you recover between each of the activities. If you go straight from exercising to hyperventilating, you're not going to get the same result. So make sure you are fully recovering before you do the second activity. So that is for the holding your breath part. The second part of the control of respiration is an activity that I would encourage you to do, where basically you are going to be making a home brewed pH indicator. To do that, you need some type of pigmented fruit or vegetable. What works best is red cabbage. So if you have red cabbage or have access to a store and can purchase red cabbage, then I would encourage you to do that. However, again, because we are at home, because we are COVID, because I know different people have different types of issues and concerns, I am not going to require that you actually do it. If you are not comfortable getting to the store or purchasing the additional cabbage for any reason or anything along that, you are not required to do this activity physically. But if you are not able to do it physically, then what I do want you to do is complete it and answer the questions as a thought experiment. All right. No one should write down, I was not able to get red cabbage or red pigmented grapes or anything along those lines, so I couldn't do that. That's fine. If you can't do it, if you can't get the stuff or don't want to do it, that is fine as well. But I want you to do the activity as a thought experiment, explaining what you think will happen. So I definitely still want you answering the questions. I definitely still want you to do it. It is fun if you want to go and do it. It's if you've got kids, you can get them to do it along with you to see how it works. So it is a fun activity, I think, worthwhile to take the time to do. But if for any reason you're not able to, that is totally acceptable, totally fine. Just please do it as a thought experiment. Do not just write unable to do or something like that on it. You will not get credit for it. It's okay if you don't do it, but write out your answers as a thought. Explain what you believe would happen and why you think it would happen. All right. That is that. That's the only tricky part on this. You have all the regular stuff. Again, Wednesday, the 14th, PhysioX is due, 3 activities, so 3 lab reports for that. Another unit review for the urinary system, our first urinary physiology activity, the Labster. Remember, you need to complete the whole activity and get it 80% correct, which reminds me, remember, if you don't get it 80% correct the first time, you are welcome and encouraged to do it again. If you do not get it 80% correct, you're not going to get full credit for it. So make sure that you go back and do it again so you get your full 20 points. And last but not least, thinking of points, your lab and lecture exams were graded last week, and I posted those grades for you so that you could see them and some of the basic concept comments that I had. Although there's a couple of you that I put comments in that have not responded to, so please make sure that you read those comments if you need to address those, that you speak with me about them. And that after class today, I will release the exam so you can actually see the questions, see your answers, see where you got your points and where you didn't get your points. And as always, those will be available for about 24 hours or so. And then you will be able to view it during those times, especially for those of you who are still doing the not the physio X, doing the science skills activities. Part one of the activities is to evaluate your exam. So this is a perfect opportunity to be able to do that. All righty, questions on any of that? All right, a little long introduction, but again, I really wanted to emphasize with that control of respiration activity, again, to give you the warnings, to give you the caveats on that. No one is to do it if they have any kind of breathing difficulties or issues. Everybody who does it should be seated down, should be observed by an additional person while you are doing these things. Please do them safely. They're fun activities. I think it's entertaining to do, and interesting, but also please do them safely. All right, with that in our pocket, then let's move on to lecture, where, like I said, we're going to be talking about the respiratory system. So let's get up that. There we go. Perfect. All right. So when we talk about respiration and think about the processes involved of respiration, obviously the most common thing that people think of is pulmonary ventilation. What is that? What is pulmonary ventilation?
Ventilation in the lungs.
Yeah, the mechanical events of moving air into and out of our lungs. That is what that pulmonary ventilation is. Absolutely. As we've talked about before, the reason we do is to get that air into our lungs so that external gas exchange can take place. Where or what, let's think of it that way. Where does external gas exchange take place? It's an exchange, so it must be between two structures. What are the two structures where this external gas exchange takes place?
The alveoli and the pulmonary capillary beds.
Absolutely, excellent. So it is between the pulmonary capillaries and the alveoli. Excellent, excellent. Right? We could also think of this gas exchange as taking place between two mediums. So what are the mediums that this exchange is taking place between? Between what two substances?
Blood in the atmosphere.
Exactly. Well, yeah, absolutely. So between blood and I would say air as opposed to the atmosphere, but you get the right idea. Absolutely. So it's between blood and air. Again, so it's the blood in the pulmonary capillaries, it's the air in the alveoli. So you can think of it, it is either of those two ways. We are exchanging between the capillaries and the alveoli, the structures where it's taking place, or the mediums, the substances where it's taking place. And let's actually sneak this over here for right now so we can see that. Oops, oh, didn't mean to do that. There we go. Excellent. Of course, once that exchange takes place in the lungs, then we need to transport that gas throughout the body. Is that really a process of the respiratory system? No. What is responsible for that gas transportation?
Cardiovascular.
Yeah, exactly. The cardiovascular system. And of course, then we have our internal gas exchange takes place. And we already know the trick to this. There is going to be a where for this, and there is going to be the what mediums for this, that is these exchanges are taking place. So let's do the where first. Where does internal gas exchange take place?
Capillary beds and the tissues of the body.
Excellent. I like that. So definitely the tissues of the body. However, we can probably be more specific than capillaries. What kind of capillaries? Since we said pulmonary capillaries before with external gas exchange, we should probably specify these. How would we identify these capillaries?
Somatic capillaries, end organ capillaries.
About systemic capillaries, right? So systemic capillaries, I like that. Although there's others work as well. Excellent. And then of course, then what would the two mediums be?
Blood in the cells.
Okay, so definitely blood. The blood is in the capillaries, but what is it that in the tissues? Is it really the cells that are directly receiving the gases? That is the goal, is to get them to the cells. But when the exchange is actually taking place at the capillary, it's between the blood and what?
Interstitial fluid.
There we go. Daniel, you have been missed. Excellent. Excellent, excellent, excellent. Spectacular. So there we go. So we have all of these what's and wheres for the different types of exchanges and stuff that are taking place, and all of the events that are really involved in the gas exchange. And of course, the whole reason for this is our cellular respiration. And someone remind me again what cellular respiration is?
The aerobic metabolism of oxygen, glucose into ATP.
There you go. Absolutely. Well, close. We definitely are using glucose and we're breaking it down completely by oxygen. So we're using that glucose, that oxygen to completely break down the glucose. What we get as a result of it is energy. And you are correct. We are going to use that energy to make ATP. And of course, there are a couple waste products of this as well. What are the waste products of this as well?
CO2 and water.
Barbaric acid.
There you go. Oops. Absolutely. And again, that's the whole reason we do. It's because our cells need the oxygen to make ATP, and they produce carbon dioxide as a waste product, which is then released from the cell. And of course, where does this take place? This complete breakdown of glucose?
Inside the cell, specifically the mitochondria.
There we go. Excellent. In the mitochondria. So there at the mighty mitochondria, as we've talked about, those, remember, are our powerhouses of the cell. We are breaking down glucose with oxygen to produce ATP. That's cellular respiration. Right. So when we talk about respiration, all of these are processes that are important for respiration. However, when we talk about the respiratory system, are all of these necessarily the responsibility of the respiratory system? No. Which ones are the ones that are specifically the functions of the respiratory system?
1 and 2.
Exactly. 1 and 2. So when we talk about the respiratory system, all of these are processes involved with respiration. But when we talk about the respiratory system is going to be involved in our pulmonary ventilation and our external gas exchange. Excellent, excellent, excellent. So again, here's a pretty picture showing all of these processes, showing both the systemic capillary and the interstitial fluid where the exchange is taking place in the tissues, our pulmonary capillary and the alveoli where that external gas exchange is taking place, transportation through the heart, and then of course the cells are doing that cellular respiration. And again, as we've talked about, pulmonary respiration, or what we call ventilation, is a function of the respiratory system. And what specifically is responsible for pulmonary respiration?
The diaphragm, the lungs, the airway passages, and the intercostal muscles.
Okay, I like that. Absolutely. The skeletal muscles are what are responsible for actually the changes that take place. Yes, you need the pathway as well for the air to move, but that the actual mechanical events that take place are the results of us controlling skeletal muscles, including the intercostals, including the diaphragm, and other muscles as well, which I'm sure you remember from 430. So we will definitely talk about those. Oops. As we talked about, that external gas exchange is that gas diffusion between the alveoli and the pulmonary capillaries. And what we didn't talk about is which gases are involved in this. Well, we kind of did. We know that it's oxygen and CO2. But in our external respiration, what direction does oxygen go?
In?
So into what, though? We want to be more precise than that.
Into the bloodstream?
Okay, excellent. So again, we can think of it as the medium that is entering into the blood, or remember, we could think of it as the structure. Right into the capillary. And which way does CO2 move?
Out through the expelled air?
True, excellent. So into the air, right? Or into the alveoli. All right, notice that's why we can't use terms like in and out. I mean, I guess technically we could say in and out of the body. That would work because this gas exchange takes place at our what we call a respiratory membrane. And that membrane, as we know, you must pass a membrane to get into the body. So I guess technically we could say into the body and out of the body. But remember, we want to think in terms of either the mediums or the structures that are involved in this. So that's a good way of doing that. But you're also right. We could do it the other way as well. For instance, for CO2, we could say out of the blood if we wanted to really emphasize the fact that it is moving in the opposite direction of oxygen. So we could do that as well. So you can use terms like in and out, but you want to be really careful about using those terms in and out and being more specific in that. Excellent. Of course, as we talked about, internal respiration is between that systemic capillary and the tissues. And so again, we want to talk about the way and direction that these go. Oxygen and our CO2. So which way does oxygen go with internal respiration?
Would more in be a correct answer?
Well, now here because this is all inside the body, so that would definitely be tricky. So again, we want to think of in terms of the structures, we want to think of in terms of the medium. So oxygen is going from what medium to what medium, or from what structure to what structure?
Going from blood to interstitial fluid.
There you go. Or from the systemic capillary into the tissues, right? We could do it that way as well. So again, you could say out of the blood into the interstitial fluid. Again, all of these are fine ways of saying it. And then which way does our CO2 go?
From the interstitial fluid into the blood.
Right. They're always going opposite directions of each other. Excellent, excellent, excellent. And then, of course, we already talked about cellular respiration, so I won't bother doing that again. Again, occurring at the mitochondria, using oxygen to completely break down glucose, and getting CO2 and water as a waste product. All right, spectacular. So now we have an understanding of what respiration really, really means and which of these processes we talked about are involved in the respiratory system. So that makes our functions pretty simple and straightforward, right? Obviously, our functions of the respiratory system are to move air, that mechanical ventilation of the lungs. But it's also more than just moving the air. One of the things that we talked about in the digestive system is you could put a straw up your nose and get milk into your stomach that way. Does that mean that your nose is part of the digestive system just because it's a path that milk can use to get to your stomach? No, of course not. And by that same token, while anybody who's ever had a cold survived it because they were able to move air into and out of their lungs through their mouth, right? Your mouth, your oral cavity is not a part of the respiratory system. Yes, it is a passageway that can be used for air, but respiratory system's passageway is much more than just a conduit. Its job is threefold. Yes, we want to move the air to the lungs, but while we do that, we want to clean the air, because after all, right, if you grab this chunk of Sacramento air right here, even though you can see through it, is this necessarily the most cleanest stuff in the whole wide world? No, absolutely not. So we don't want to just take this and dump it straight into the lungs. We want to clean it. We want to filter it first. We also want to warm the air. Why do we want to warm the air? What's the advantage of warm air?
If it's warm, that means it has more energy, so it probably readily diffuses faster.
Exactly. And that's really one of the interesting things about gas exchange, right? How long can you survive without oxygen?
I think like 6 minutes.
Yeah, so not a very long period of time, right? Or how long can you survive without getting rid of CO2 from your body? Probably even less. As we'll learn, CO2 is actually even more important. When you're drowning underwater and you're trying to hold your breath for as long as you possibly can, is it the loss of oxygen that causes you to breathe in that water when you're underwater? In most cases, it's actually a buildup of CO2. So CO2 is actually, as we'll learn, more important in determining your rate of respiration than oxygen levels are. So with this gas exchange, it is vitally, vitally important to get oxygen into our body and get CO2 out of our body. And yet the entire process is passive. Someone remind me again what passive means?
Doesn't require energy.
Yeah, specifically ATP. It does not require ATP. So as Daniel pointed out, by warming the air, we are able to actually get those molecules to move more quickly. And as they move more quickly, it increases the rate of diffusion. Lastly, not only does it filter the air, not only does it warm the air, but it also moistens the air, because that gas exchange that is going to take place occurs more readily through water. So by moistening the air and putting moisture in the air, it is again going to help to facilitate and make that passive diffusion of gases happen more readily. So the goal of our respiratory passageway is not just to move it to our lungs for the exchange to take place, but to warm it, to moisten it, and to clean it. And the oral cavity just isn't specialized to do those things. And that is why it's not considered part of the respiratory system. All right, excellent. And we'll see those specialized structures and talk about them as well. Excellent. So those are our primary functions, but obviously our respiratory system is involved in other things as well. It provides us with our sense of smell. It plays a vital role in helping to regulate the pH of our body, helping to maintain the appropriate pH of our blood and and therefore, again, remember, whatever happening in our blood happens in our interstitial fluid. So maintaining that appropriate pH, make sure that our proteins are working properly. And then, of course, think how horrible my lectures would be if I wasn't able to use my respiratory system to produce sound. Right? Sitting here for 4.5 hours listening to me talk is pretty bad, but sitting here for 4.5 hours watching me type on the computer and reading it would be even worse. So that ability to produce sound is a vital function of the respiratory system as well. All right. Questions on that? All right, excellent. Not surprisingly, like most things we've talked about in the body, when we distinguish our respiratory system, we can divide them one of two ways. We can divide them anatomically, and we can divide them functionally, right? Anatomy and physiology, structure and function. If we were to do a structural division, of the respiratory system, we would divide it into two halves, the upper and lower respiratory system. If you notice and look closely, you can see that that division takes place right at our friend, the epiglottis. Remember, our epiglottis is what folded over during deglutition for that swallowing reflex. So basically everything from that epiglottis up is considered the upper respiratory system, and everything below it is considered the lower respiratory system. So that is structurally how we are going to divide it. And quite frankly, it's a stupid way to identify and distinguish the parts of the respiratory system. I'm sure there must be some meaningful reason why they do it this way, but I don't happen to know what it is. And I think it is silly. I think it makes much more sense to think of the respiratory system in terms of their function. And when we talk about the function of the respiratory system and talk about a functional division, I think that is much more meaningful. We distinguish the parts by what they do. And if we do that, then what we have are two main parts to the respiratory system. We have our conducting zone, and our conducting zone is the part that we just talked about. It is responsible for the movement, the filtering, the moistening, and the warming of the air. And then the respiratory zone is where the external gas exchange takes place. That respiratory zone occurs, not surprisingly, at our respiratory membrane. And that respiratory membrane is basically comprised of the walls of the alveoli and the walls of the pulmonary capillary. There. I think that is a much more meaningful division. Clearly, all of the respiratory system, upper respiratory system, is involved in conducting, but so is most of the lower respiratory system. So again, I'm sure there's a meaningful reason why for some people, some things are done that way. But I think functionally, for me at least, makes much more sense. But do be aware that there are the two ways to divide it. Here we get a closer look at the anatomy, and we can see many of the structures that we are already familiar with. We have already talked about the pharynx, and the pharynx has three main regions, as we've talked about: the part associated with the posterior part of the nasal cavity, nasopharynx; the part associated with the posterior part of the oral cavity, the oral pharynx; and the part across from the epiglottis and the larynx, which is the laryngopharynx. We've talked about those as well. We've talked about the pharyngeal tonsils, which is at the superior part of the pharynx. Palatine tonsils located in the fosse between the arches of the palate. We've talked a little bit about the epiglottis and a little bit about the larynx, so we'll talk about it a lot more. We've talked about the nasal cavity a little bit, where we have these three bumps inside the nasal cavity. What are these three bumps in the nasal cavity collectively known as again?
The turbinate. turbinae, turbine.
No, Allison's got it. Concha. Absolutely, these are concha. Absolutely. And remind me again, this first concha up on top is the superior nasal concha. Is it a bone or a bone feature? Is it a individual bone by itself, or is it a piece of another bone?
A feature.
It is a feature. Feature of what bone? No, you learn these bones back in 430.
Ethmoid.
Ethmoid, excellent. Middle nasal concha, bone or bone feature?
Feature.
Which bone?
Eth.
Ethmoid, excellent. Inferior nasal concha, bone or bone feature?
Bone.
Bone, absolutely. There are two of our paired facial bones. Inferior nasal concha, excellent. So we have those, we've talked about that. Let's take a look at a different view. Here we actually see it in an actual cadaver. We can see these structures as well. And actually, while we're here, we'll come back to this one real quickly. Actually, no, because I think it shows it here. We'll show this as well. Notice with the conchas, one of the things that you'll notice is that there are spaces underneath them. The spaces underneath the conchas are known as the meatuses, and these are passageways for the air. There also, although you can't see all of them in this view, there are two that can be seen here. There are a total of 4 paranasal sinuses. Remember, a sinus is an air-filled space. These are paranasal, so they're associated with the nasal cavity. And anybody remember the 4 bones that help to form the paranasal sinuses? Have sinuses in them that form them. I'll give you 2 big hints. One can be seen right here. The other technically can't be seen, but we know it's in this bone right up here. So what are the 4 bones that have sinuses in them?
Frontal, ethmoid, maxillary, and sphenoid.
Excellent. Frontal, ethmoid, maxillary, and sphenoid. Spectacular. There are those 4 bones that have those sinuses associated with the nasal cavity. These paranasal sinuses, these concha, the goal of all of these structures, rather than just having it a straight tube back, we have all these structures, all these spaces, and their job is to produce turbulence. What's the advantage of producing turbulence? Well, yeah, absolutely. Part of it is the warming. When you produce turbulence, you're swirling the air around. It's not going straight down into the lungs. So not only does that give it time to come in contact with the very closely associated blood vessels for warming, but it also comes in contact with all the mucus of our mucous membrane for filtering and warming and moistening. Right, so this gives time to process the air. That turbulence keeps the air in the nasal cavity for a longer period of time so it can be processed. So it can be warmed, it can be filtered, it can be moistened. Absolutely. Excellent, excellent, excellent. So nose is the only external respiratory structure. Air enters through the external nares into a region called the vestibule. And I like this cadaver picture because you see this very, very nicely. We don't see the nostril because the septum's in the way, but what we do see is that it leads into this pocket in this space here. That is the vestibule. Those are those lateral structures, right? That's where the pinky goes when you're trying to pick a booger, right? Notice in there, you'll notice some protective structures. Those protective hair-like structures help to stop insects, large particles from entering into the nasal cavity. right? Those are those nose hairs. Of course, we're absolutely positively not going to get away with terms like nose hair on the exam. So what is the appropriate anatomical term for nose hairs?
Vibrissae.
There you go, vibrissae. Absolutely. They are vibrissae. Whisker-like hairs that provide that protection to the entrance of our respiratory passageways. Excellent, excellent. The interior is that nasal cavity, and it is lined by what type of epithelium? Well, let's start here. What type of membrane? We'll start easy. What type of membrane lines our nasal cavity?
Mucous membrane.
There we go. Mucous membrane. And of course, all membranes are comprised of epithelial tissues on top of connective tissues. We'll start easy. What is the connective tissue that lines a mucous membrane?
Areolar.
There we go. And as we just learned in the digestive system, based on its location, what do we call this areolar connective tissue? That is part of a mucous membrane.
Lamina propria.
There you go. Just like we saw in the digestive system. However, unlike the digestive system, our nasal cavity has what epithelial tissue that lines it?
Olfactory epithelium.
It does have the olfactory epithelium up there at the superior part of the nasal cavity that allows us to sense smell, but it isn't the primary epithelial tissue. And it's not a non-keratinized stratified squamous. That's what we have in the oral cavity. This is the one that you learned way back in 430. It took you forever. It was like an alphabet soup phrase that you had to learn. You were hoping you would never have to use again.
Ciliated pseudostratified epithelium.
There we go. Close. Ciliated, pseudostratified. Oops. Columnar. Epithelium. There we go. A ciliated pseudostratified columnar epithelial tissue. Absolutely excellent, right? With those cilia that take the mucus and move it to the back of the, really to the nasopharynx, so it can either be swallowed or spit out. Excellent, excellent, excellent. So we have that mucous membrane with a ciliated pseudostratified columnar epithelial tissue in it. Right? Unless you snorted massive amounts of cocaine in the 80s, you should not be able to put a handkerchief up one nostril and pull it out the other, right? Although I guess now if you have a septal piercing, right, and take it out, you may be able to do that. So I may have to work on my analysis there. But the nasal cavity is divided in half by a skull feature. And what is that skull feature that divides the nasal cavity in half? Pretty sure I just gave it away when talking about the piercing.
The septum.
Yeah, exactly, the nasal septum. But what I didn't give away were the bones and bone features that form it. See, I told you way back in 430, the stuff we were learning there wasn't just random, right? This was crap you were going to have to use again. So what were the bones and bone features? Obviously, there's connective tissue. Obviously, there's cartilage that helps to make the nasal septum. But what were the bones and bone features that made up the nasal septum? Excellent. Perpendicular plate, which is a bone feature of what bone? Whoops.
Maxilla.
No, it wasn't the maxilla. Perpendicular plate was a bone feature of what bone?
The ethmoid bone.
Ethmoid. Excellent. And what was the other bone? That plowshare shaped bone right on the midline of the body?
The vomer.
There you go. Excellent. Right, so we have that vomer that helps to divide the nasal cavity as well. Excellent. So we have that nasal septum. One of the interesting things at any one time, if you close one nostril and try to breathe through both sides, you may notice that you're not able to move air as equally through both passageways of your nasal cavity. The reason for this is, as I talked about, the nasal cavity is specialized for the processing of the air. And if it is too dry or not warm enough, there is actually erectile tissue, similar to the erectile tissue we see in the penis and in the clitoris, in the labia of the female, that can actually engorge with blood to restrict passage of air through one side versus another. And so what will actually happen during the course of the day is you will have this enlargement of that tissue restricting airflow to help to maximize the efficiency. Many years ago, and I'll be honest, I don't know if it was ever if they ever did this experiment again and tested the validity of this study. But many, many years ago, gosh, it was probably like 20 years ago, there was a research paper that was done where some researchers actually looked at cognitive function of individuals as they were able to move air through one side versus the other. And they claimed, because again, you know, if you think about it, especially with the olfactory epithelium so close and the cribriform plate and the olfactory foramina, they claimed that, for instance, individuals were better at mathematical problems when they were able to breathe through one side of their nose versus the other side of the nose or something like that. It was a really interesting study. I don't know if it's ever been validated, if anybody else has ever been able to recreate those results, but at the time it was very, very interesting, the premise to think about. So yeah, so absolutely. We have that divided nasal cavity. And again, to beat the dead horse, the mouth is not part of the respiratory passageways. All right, we talked about the paranasal sinuses, and here we have the pretty picture that shows them from your textbook. Again, here we see the maxillary sinuses, basically to the lateral sides, inside of that big solid maxillary bone. What we think of is, for instance, you know, those apples of your cheek right there. It's actually a space inside of there. The frontals are the ones that are up here at the top. And then, as you can see along the midline, we have the sphenoid sinus and the ethmoid sinuses. And again, remember, these sinuses are lined with mucous membranes. And when those mucous membranes become congested, there can be an increase in mucus production, causing swelling, causing pressure, causing pain in those areas that we typically associate with, you know, sinus pain or sinus infections when those occur. Right? Every time my wife speaks, for instance, I have a massive increase in mucus production in these frontal sinuses, and I just get this throbbing pain in there every time I hear her voice, right, as a result of that. So I'm sure many of you have experienced that. I don't know if any of you have ever had problems with oral surgery before. One of the things that happens, our friend, the trigeminal nerve, actually comes into this maxillary sinus, where it then goes to the maxillary, goes to the mandible, and goes to the olfactory. So there's this massive neural structure here. If you've ever had a maxillary sinus infection from like a root canal that went bad or something, it is incredibly painful. Anyone here ever had maxillary sinus infection before? No one willing to admit it. It can be incredibly painful. It's a good thing if you haven't, because it is not a fun thing to experience. But there's this massive nerve structure located in that maxillary sinus that can cause tremendous amounts of pain. And again, the point of these sinuses is to produce that turbulence, swirl that air around so that it can be warmed, so that it can be cleaned, so that it can be moistened, to maximize the health and efficiency of our gas exchange. And I love this picture, a posterior view of a coronal section from your book, which I think it does such a great job of showing here. I need a thinner line for this. Here we see that nasal septum down the midline. Here we see the superior nasal concha, middle nasal concha, inferior nasal concha, and then the meatus between them. Notice we also see our frontal sinuses. We can see our maxillary sinus very, very nicely. The tiniest bit of our ethmoid sinuses here to the side. And then the only thing we're not seeing, obviously, is the sphenoid sinus because it's much further back. But this is a great, great view. And again, it just shows that it's not just a hollow tube pathway. There's all this structures, all this turbulence to process our air. Also, even though we didn't talk about it, we know, of course, that lining the mucous membrane of here are a lot of very superficial blood vessels. What's the advantage of having all these superficial blood vessels inside the nasal cavity?
To warm the air.
Exactly, absolutely, right? Remember, blood is 2 or 3 degrees warmer than our core body temperature. So having all these blood vessels close to the surface are able to radiate off their heat and warm that air as it comes in. Of course, what's the downside of having so many blood vessels so close to the surface in the nasal cavity?
Horrible nosebleeds.
Yeah, exactly. Irritation from dust particles, fingers getting stuck up there, things, getting pressure concussed into it can cause those nosebleeds to hurt as a result of it as well. Now, our air exits hopefully I have a picture. Let's see how clever I am on this. Otherwise, we'll hunt one down. Exits the nasal cavity by an opening called the internal nares. The internal nares, and let's see just how clever I am. I'm not clever enough. All right, let's stop for a second. Here, here. What's wrong? There. There we go. Now I'll be clever in the next class. You know I'm not clever, but the next class will know that. Excellent. So here we see our friend the skull. Of course, here we have that hard palate that we just finished talking about in the digestive system. And here we can see formed from the 2 palatine bones and the vomer posteriorly, the ethmoid, well, medially, I should say, and the ethmoid superiorly, we can see these 2 openings that are the internal nares. So this is the bony structure that the air passes through to get back to the pharynx. So there we see that pretty picture of that opening. Excellent. We already talked about our respiratory mucosa, a ciliated pseudostratified columnar epithelial tissue in our nasal cavity. Where else do we have that ciliated pseudostratified columnar? And I'm just going to abbreviate it here, ciliated pseudostratified columnar epithelial tissue in the nasal cavity. Where else do we find that ciliated pseudostratified columnar epithelial tissue? All right, well, you guys figure it out. I guess I will write it out.
The alimentary canal.
No.
The trachea.
There we go. Yep, and the trachea. Again, remember, the goal of these cilia are to take mucus and move it, collect the mucus and move it. In the nasal cavity, those cilia take and move that mucus that's captured the dust and the debris to our nasal pharynx, so it can be swallowed, it can be spit out. And in fact, I guess we should throw that as well. Not only the nasal cavity, but the nasopharynx is also lined with a ciliated pseudostratified columnar epithelial tissue, as we talked about. But it's also located in the trachea. right? And the goal of it in the trachea is to form what we call this mucus elevator, where the job is to take mucus that is capturing dust and debris and move it away from the lungs back up to the larynx, where it can either be, again, spit out or swallowed. All right. As we talked about, these cilia are delicate. They're sensitive to temperature changes, right? They can be overworked if you're overproducing mucus, right? And if you happen to be a smoker, and this includes vaping, right, that smoke can damage those cilia. And so now when that mucus is produced in the trachea, if your poor little cilia can't move it up, how do we move that mucus?
Cough reflex.
There you go. Exactly. You got that great smoker's hack to get that mucus up out of the trachea, which is one of the reasons why if you have a cold and you're a smoker, you have to be really, really careful about taking cough suppressants. Because if you no longer have a cough reflex and your poor little cilia have been damaged and aren't working anymore, what happens to all that mucus?
Collects.
Down.
Yeah, slides right on down into the lungs, dramatically increasing the likelihood of infection, COPD, pneumonia, all sorts of other conditions that can occur because of that. Excellent, excellent, excellent. So we know that's in there and we know its function. However, as we talked about, we know that the oral pharynx and the laryngopharynx are not lined by ciliated pseudostratified columnar epithelial tissue. So what kind of epithelial tissue are they lined by? Again, we just finished talking about this in the digestive system, so we should know this easily.
Non-keratinized stratified squamous epithelial tissue.
Yep, see, you were right earlier, Allison. You just were early, that's all. Absolutely, a non-keratinized stratified squamous epithelial tissue. Because again, that region is a passageway for, yes, air, but also for food and water. Now, once we start getting down the trachea to the bronchi, it is going to again start with a ciliated pseudostratified columnar epithelial tissue. But is it going to be lined by that, do you think, the entire way down through the respiratory system? Well, the answer is no, because I'm asking the question. If the answer was yes, then I would be making a point of emphasizing it. It's going to transition first to simple columnar, then to simple cuboidal, until finally we get to the alveoli. When we get to the alveoli, where that passive gas exchange is going to take place, what type of epithelial would we want there to maximize the efficiency, or more specifically, decrease the distance that those gases have to travel?
Simple squamous.
Squamous.
There you go. So there's quite a transition in the epithelial tissues of our mucous membrane here in our respiratory passageway on the way down. Of course, all of these are part of the mucous membrane. So again, they have an epithelial tissue sitting on top of an areal arc connective tissue that is going to be a lamina propria, all the way down to the alveoli where gas exchange is going to take place. And here we see a pretty picture of the ciliated pseudostratified with their mucus goblet cells in here, again, in the cilia and all the fun stuff going along with that. We did that. We did that. We did that. We mentioned this briefly, but I'll remind you again. The olfactory receptors are bipolar neurons. That are located, as someone mentioned earlier, at the superior part of the nasal cavity in our olfactory epithelium. These olfactory bipolar neurons actually form what special structure?
A cranial nerve.
Yep, which cranial nerve?
One.
Yep,
Yeah.
cranial nerve 1, absolutely. Which again gives us our sense of smell. And even though I said it earlier, I'll remind you again, it passes through what bone feature? As its skull exit?
The cribriform plate?
True, it is the cribriform plate of the ethmoid, but what are the actual holes that they pass through, the holes in the cribriform plate that it passes through?
Foramen of some type.
Yeah, in fact, they're the foramen of the olfactory system. They're the olfactory foramina. Our foramina is just the plural of foramen, those many little holes passing through there. And in fact, again, because I didn't set it up ahead of time, but let's do it here since I have this up. That one works. There we go. Here's that ethmoid, remember with the cristogali. Here are those cribriform plates, the big flat surfaces where the olfactory bulb sits, and the olfactory foramina are the little first cranial nerves, those bipolar neurons that pass through the cribriform plate into the superior part of the nasal cavity, providing us with our sense of smell. Right? As those of you who took me for 430 know, my sister once dated a young man who on a New Year's Eve got incredibly drunk and fell down a flight of stairs. And as he fell down a flight of stairs, as his brain was rattling around inside of his skull, his olfactory bulbs tore off of his cribriform plate. And when he woke up the next morning, what could he no longer do? Yeah, he lost his sense of smell. But as I've said many times, it does have a happy ending. She broke up with him. All right, excellent. True, he was very lucky he could walk. And in fact, while she did break up with him, he was one of the groomsmen at her wedding. So, because she ended up marrying one of his best friends. Yeah. My sister grew up in Kentucky. No, I'm just kidding. All right, excellent. So, as the air leaves the nasal cavity, it enters into the pharynx. Again, as we know, that pharynx is a muscular passageway that has those three regions, the nasopharynx, the oropharynx, and the laryngopharynx. And again, as we know, nasopharynx is air only and is lined with also the ciliated pseudostratified columnar epithelial tissue, whereas the oropharynx and laryngopharynx is air and food and drink. And since it's air and food and drink, it is lined with a non-keratinized stratified squamous epithelial tissue. There we go. Excellent. So again, I think we'd already said all of that. So again, no reason to spend too much time emphasizing that. And more pretty pictures showing what we already know. Excellent, excellent, excellent. All right, from there, we will move on to the larynx, but I think this is a good stopping point because we have a fair amount of anatomy we need to talk about. So let's actually stay here for now. Seeing as this is the most talking I've done in a week already, just 1 hour in, then let's go ahead and take our first break. I will get some coffee, and we will meet back here at 9:15 to restart. And I will remind myself to start the recording at that time. All right, any questions before we take our first break? All right, see you guys in 15 minutes. All righty. Hopefully we are all caffeinated and ready to roll. So we have so far been working our way through the anatomy of the respiratory passageways, starting through the nasal cavity into the pharynx, which we are familiar with, and that is going to bring us to our larynx. We talked about the larynx. We know the larynx is what elevates during the swallow reflex that we talked about, deglutition. We know about the epiglottis. We've talked about that. We felt the Adam's apple, but now I want to take more of a close look at the anatomy of it. Of course, it is lined with a ciliated pseudostratified columnar epithelial tissue. It is also a passageway just for air. However, there are two folds in the mucous membrane, and you can see them here and here. Why are those so small? There we go. You can see them here. Oops, hold on, let's just clear all of this. That one there and that one there. One is superior, one is inferior. These are what people commonly refer to as the true and false vocal cords, but the appropriate names for them are the vestibular fold, which is the superior one, and the vocal fold, which is the inferior one. And I think I have another view of these. I do. Let's cheat and go ahead. There we go. When you look at these from the superior view, as we're looking down into the pharynx, we can see that the vestibular fold, as we said, is superior. Notice it's also a little bit more lateral. This one has a very sensitive mucous membrane on the surface of it, so that if any dust and debris come in contact with it, it causes a coughing reflex where you rapidly force air out of your lungs and helping of expelling whatever that irritant is. Deep to it, but also more medially, is a much more inflexible fold, that is the vocal fold. This vocal fold is avascular, as you can see here, and it has the ability to vibrate. And the vibrations of it are what produce our sound. So we have those two folds. We see them here from a superior view. And we'll talk about them more in a minute when we come back to that. But here we see them, again, in their location inside of the larynx this way as well. Now, the larynx is comprised of a total of 9 pieces of cartilage, 8 of which are rigid hyaline cartilage. And their function is to basically keep the airway open. Right? Remember, our pharynx, our esophagus were muscular tubes. When you're not eating, those things basically collapse in on themselves, decrease in their size, decreasing their lumen. However, our airway, we want to constantly keep open. What's the first thing you do when you go to blow up a balloon?
Inhale.
Okay, true. Inhale. What do you do to the balloon?
Hold it by the
Your
tip.
mouth.
Stretch it out.
Okay, so you grab it, you hold it to your mouth. Is that really what you do? You reach in the package, pull out a balloon, and instantly put it in your mouth. That's what everybody does.
Oh, you make sure it's like round. It's not crumpled. Does that make sense? I don't know.
Yeah, you stretch it out. Why do you stretch it out?
So you can make it bigger.
True. So one thought might be so that it helps to loosen the elasticity to make it bigger. Turns out that's not actually what you're doing. When you stretch it out, what you're doing is most of the time when it is inside of that package and it has been collapsed, the two sides come in contact with each other. And when they come in contact with each other, how easy is it to get the air in there and force the two sides apart? Not. So when you stretch it out, basically you separate the two sides from each other, and that makes it easier for you to put air into the balloon, right? You are breaking up that surface tension. Breaking up surface tension is really, really hard, right? And when there's moisture involved, it's even more challenging. Think of it in terms of, and summer's right around the corner. So you're going to be sitting in the backyard with your big glass of lemonade on your glass patio table. And when that condensation on the outside, that moisture, that water gets between the glass of your cup and the glass of your table, how easy is it to pick that glass up? Yeah, it suctions to it because of the surface tension of the water. And so we don't want that happening to our airway. We don't want our airway collapsing and us having to force it open. So having this rigid hyaline cartilage helps to keep the passageway open. Of course, as we know from swallowing, they're not all hyaline cartilage. The epiglottis, remember, is that elastic cartilage, that rubbery structure that allows it to fold over the top. Here, we peel away most of the pesky person and we can see more of the anatomy of the larynx. Notice, as I mentioned on the previous page, the largest chunk of hyaline cartilage is a structure known as the thyroid cartilage. Of course, why is it called the thyroid cartilage? Well, because if you remember, the thyroid gland, that two big lobe with the isthmus gland, sits here around, wraps around the trachea, just inferior to the thyroid cartilage. So that's where it gets its name. That prominent part that sticks out, the Adam's apple, as we commonly refer to it as, is known as the laryngeal prominence. Laryngeal prominence cartilage is stimulated by androgens, testosterone. So typically when a male reaches puberty, this cartilage thickens, and typically males have a more pronounced Adam's apple than females do as a result of that. Now notice, while it is by far the largest chunk of hyaline cartilage, if you look at it here from a side view, and more importantly here from a posterior view, you see that this large shield-like structure only goes 3/4 of the way around. It is actually open in the back. In fact, if you look closely, you will notice that there is only one piece of cartilage for the larynx that actually forms a complete circle. Notice here it is smaller in the front, larger posteriorly, but it is the only completely circular piece of hyaline cartilage, and it is what connects the larynx to the trachea, what is known as the cricoid cartilage. So we have the thyroid cartilage, we have the cricoid cartilage. Those are two individual cartilage pieces. Notice from this posterior view, we can see that there are two paired cartilages. One is this pyramid shape cartilage piece that we see here, known as the arytenoid cartilage, and here we see it from a side view. And notice on top of it is this tiny little corn kernel piece known as the corniculate cartilage. Again, why? Remember, oh, what does it connect?
Well, you said that the cricoid cartilage goes all the way around and then it connects something posteriorly?
To the trachea. No, well, no, what I said is that it's notice that it is larger posteriorly. Notice it's much thicker, much larger posteriorly than it is anteriorly. But what it does is it connects the larynx to the trachea underneath. So it caps the trachea. So yeah, so it basically is the base of it that sits on top of the trachea. All right, excellent. So there are our 8 pieces of hyaline cartilage that make up our larynx. Questions on that? No one has a question? I have a question. How many pieces of hyaline cartilage did I say were here?
8.
How many have I identified? True, 9 with the epiglottis. That is true, 9 with the epiglottis, but 8 pieces of hyaline cartilage. And how many pieces of hyaline cartilage have we identified? Well, let's count them. Thyroid cartilage, cricoid cartilage, 2 arytenoid cartilages, and 2 corniculate cartilages. How many is that? I know it's Monday morning after spring break, but hopefully you guys can count to 6. We are missing 2. And that's because we don't see the full anatomy of the larynx here. Notice what we are seeing here are the cartilages. We are seeing some of the ligaments that attach this to other things, like the cryothyroid ligament. Here we see the thyrohyoid ligament that we can see here. Notice we also see the hyoid bone. Anybody remember what's so special about the hyoid bone? What are the 2 things we know about the hyoid bone?
The tongue connects to it. It anchors.
Absolutely, it is the movable base of the tongue. What's the other really special thing about the hyoid bone?
It's floating. It's not connected to other bones.
Exactly. It is the only bone in the human body that does not articulate with any other bone. It is held in place solely by ligaments, tendons, and cartilage, and muscle and things along those lines. Excellent. So we're seeing some of the structures here, but around these cartilages is also our mucous membrane. And notice when we put our mucous membrane in place, like we can see here, what we can see is within the wall of the mucous membrane. Well, let's talk about that wall of the mucous membrane first. Notice the mucous membrane lines the surfaces. However, you will also notice that there is a fold in the mucous membrane that basically connects the epiglottis to the arytenoid cartilage. Anybody have an idea what that fold in the mucous membrane might be called?
Arytenoid muscle.
No, well, that's a good guess. Now, if I had my study guide printed out, I might be able to look at that and determine what that fold in the membrane might be.
The epiglottic fold.
There you go, the aryepiglottic, because again, it goes from the arytenoid cartilage to the epiglottis. So the aryepiglottal fold, and notice in that fold is solely contained within that membrane is a small chunk of cartilage known as the cuneiform cartilage. This cuneiform cartilage helps to stabilize the aryepiglottic fold, helping to keep it open, but it is completely contained within that mucous membrane. So when we remove that membrane, we don't see that cartilage. Now, there's a couple ways we can see this, and let's again see how clever I am. Apparently, well, I'm somewhat, but let's do this. Let's cheat and go to see what CRC has done for us. There we go. That's what I want. you should remember this view of the nuchal region of the body, a posterior view of the oral cavity and the pharynx. Notice here we see that the muscular pharynx has been cut and folded open from this posterior view. Remember, we were here before because we were looking at the thyroid gland and the parathyroid glands that were located in here. But notice here we get this great view of the larynx. Notice here we have the epiglottis, that bendable piece, and here we see the airy epiglottic fold in the mucous membrane. And then notice if you look closely, there are 1, 2, 3, 4 bumps we see on the posterior part of the, and let's change colors. 4 bumps that we see on the back of this opening to the larynx. These 2 that are medial and inferior are those corniculate cartilages that are sitting on top of the arytenoid. So deep inside here, we have that triangular arytenoid cartilage and that corniculate sitting on top of it. Whereas these that are superior and are more lateral are these cuneiform cartilages that are just embedded within the area epiglottic fold. So we see those bumps really, really nicely when we look at this illustration here. There are some models, and again, I notice here again, we see those 4 bumps, 1, 2, 3, 4. The corniculates, the cuneiforms, we can see those there, but I'm hoping they have a particular model that I was looking for, and of course they don't. Shucks. Yeah, they don't have it here. Okay, I'll see if I can find it. I might even have posted it on your website. I have to look to see what's there. But there are some nice models that do a good job of showing this as well. The other place we see this nicely is here, this illustration. Here, notice with this illustration here, again, we see those 4 bumps. The 2 that are more medial are the cuniculate. The 2 that are more lateral are the cuneiforms. So again, when we look at it from the side view, that's what I wanted. There we go. We see those bumps here on the side. They stabilize that airy epiglottic fold in place. All right. So now we have 8 pieces of hyaline cartilage, 1 piece of elastic cartilage forming our larynx. Now, the other nice thing that we can see, and actually let's jump back to this picture. Notice this picture from the posterior view. We again have, oops, that's what I want. This arytenoid cartilage, which sits on top of the cricoid cartilage, has the corniculate cartilages on top of them. They actually have the ability to rotate. And so as you can see, there are tendons that are attached to it, ligaments that are attached to it. And what these things do is they have the ability to rotate. When they rotate, that opens and closes our glottis. The glottis is the opening of our airways. And so by rotating, they are able to take that vocal fold and either close it or open it. So the rotation of the arytenoid cartilages with that corniculate cartilage on top is able to open and close our glottis. If you remember, we talked about closing the glottis for that for the Valsalva maneuver, where we are forcing the air down to help in defecation, right? You closing the glottis so the air cannot escape, and so it pushes down on the diaphragm instead. So this ability to open and close the airway is by the rotation of that arytenoid and corniculate cartilage. All right. As I mentioned, there are these 2 sets of fold, the superior, lateral, And again, very sensitive and flexible one on the top is that vestibular fold with that highly sensitive mucous membrane. Inferior to that is the vocal fold, which is more medial and has that elasticity that allows it to vibrate. And as we change the tension, we can change the rate of vibration. And when we change the rate of vibration, what effect does that have?
Sound.
What about the sound? What does it change of the sound?
The pitch.
There you go. Pitch of the sound. Absolutely. When you have a higher vibration rate, what happens to the sound? Come on, any of you tuned a guitar string before? When you tighten the string and then you pluck it and it vibrates at a faster rate, what happens to the sound?
Higher pitch.
Higher pitch. Absolutely. The other way is you could force more air through it. If you force more air through it, basically you change the amplitude or the size of the vibration. And what does that change? Instead of your waves being this size, they're now this size. What's happened to your sound?
Volume higher?
Yeah, changes the volume. Changes the volume of the sound, absolutely. Right? Larger amplitude equals a larger volume. Right? This is how we can change the pitch of our voice, deeper or higher, right? Or the volume, quieter or louder, right? Now, if I were to actually take a microphone and shove it into my mouth all the way down here to these folds, when I was speaking to you, would you actually hear the words that are coming out of my mouth right now?
No, you use your tongue and your lips.
Exactly. What you would actually hear is a buzzing sound, right? That buzzing sound is what we call phonation, right? Where we are actually producing this buzzing sound. And then as Laura pointed out, we then use the muscles of our mouth, of our tongue, the bones of our teeth and our head, our nasal cavity, all of these to shape that sound into a meaningful speech, and that process is articulation. And again, most of you probably haven't thought of it in those terms, but you're aware of it. Because if I were to be able to reach in the screen and hold your tongue right now, and you were to try to speak while someone was holding your tongue, would that sound normal? Right? Or back in ancient times, you used to call people on the phone. And when you would call someone on the phone and they would say, hello, who is this? And just from that, you would say, oh, no, you're sick. What can I do for you? How do you know they're sick? Right? Because that congestion in the nasal cavity changes the pitch, changes the quality and the tone of their voice. And you can tell that they're congested just by them speaking.
So how do those microphones, like for patients with stomas, they hold the microphone to the stoma and words come out? How is that picking up the articulation?
It isn't. It is doing the articulation for you. So basically it is, I'll be honest, I don't know exactly how it works, but you absolutely have the right idea. Those machines basically take it and they form the articulation. So there, again, how it does it, I don't fully understand. I'm not familiar with that technology, but you're right. Its job is to do the articulation for them. And again, it does a decent job in that you're able to understand what they're saying, but does it sound like a normal, natural sounding voice?
No, it sounds like Stephen Hawking's voice.
Right, exactly. So while obviously it is able to manipulate the sound that is being made to make a meaningful sounding articulation, how it actually works, I'm afraid I don't know. All right. One last example. Again, this is all consciously controlled. Right now, we don't think of it that way. We don't think that you're not actually thinking about how you are contracting the muscles or tightening the muscles to be able to do that. But you can change the pitch to go deeper or to go higher as you want with that. And the other example of that is, for instance, when a male enters puberty, not only does the cartilage enlarge, but the vocal fold enlarges as well. And as it thickens, the rate at which it's vibrating changes. And that male needs to relearn how to subtly put tension into it to produce meaningful speech. And so as a result of that, not only does their voice get suddenly deeper, but do they have complete command of that voice right away? Now, what happens to that boy's voice as he enters puberty?
Cracks.
Yeah, you get that cracking of the voice, absolutely, right? Because they are not fully capable of controlling it consciously in the right way too. So they have to basically relearn how to contract those muscles to produce meaningful speech without that cracking of the voice. All right.
Generally speaking, do males have wider vocal folds than females?
Thicker, not necessarily wider, but they have
Oh.
thicker, they thicken. And so that, again, if you look at the first thing we wrote, with a thicker vocal fold, it tends to vibrate more slowly. And since it vibrates more slowly, we get a lower tone or a lower pitch to the voice in general for males than for females.
Cool.
Yep. Especially after puberty, when that testosterone causes that vocal fold to thicken. All right. All right. Excellent. So we now know the bits and pieces of the larynx. We've talked about its function. We've talked about the function of the vocal fold. What we need to do now is talk about what happens next. The air enters into the trachea. Now, one of the interesting things about the trachea is it is a single structure that goes the length, what we commonly refer to as the windpipe. But again, don't use these common terms on the air. And remember, as we talked about, there is that completely circular piece of cartilage, that cricoid cartilage that sits on top of it. Notice as we look at the trachea, we see all of these cartilage rings along the surface of it. But as it turns out, these cartilage rings are not circles. These are, in fact, C-shaped cartilage rings that don't go around the entire circumference of the trachea. In fact, we see that nicely when we look at it in a cross-section view. Notice up here in the illustration, we can see the C-shaped cartilage coming around of that ring. Here we see it as well with our light microscopy. And we can see that it doesn't go all the way around. It is open at the back. And here we can see why, because it is adjacent to that esophagus. And as we talked about in the last class, while I'm sure everybody in this class chews their food the 35 times they're supposed to before swallowing it, some people will take a big, huge bite of pizza, chew it twice, and swallow it down. The esophagus, being a muscular tube, is able to expand to accommodate that. But if you think about it, if this big, huge ring of cartilage continued all the way around, then that could impede the movement of food through the esophagus. And so that could dramatically increase the likelihood of food getting stuck. That food getting stuck could put pressure on the trachea and potentially partially or even completely close the trachea as a result of it. So you can see the benefits of having it open in the back. Now, we still want to be able to control the size of the lumen. So you will notice there is a small piece of smooth muscle that connects the two ends of the posterior part of these cartilage rings together. And any idea what that small chunk of smooth muscle might be called? If you're not 100% sure, you could always cheat and look down here.
Cricothyroid membrane.
Nope, it's the trachealis muscle. So we're down in the trachea now. It's the trachealis muscle. This trachealis muscle is a little bit of smooth muscle that can help us to adjust the diameter of the trachea. Now, with it being a big C-shaped cartilage ring, is this something that we could completely close if we wanted to? No, we're not going to be able to completely close it, but we are able to adjust the size of it with the trachealis muscle. And if you remember when we talked about blood vessels, even minor changes in diameter can have a huge effect on the resistance, have a huge effect on the flow of the air involved in it.
The trachealis muscle is only in the posterior part?
Yes. Yeah, only in the posterior part, basically connecting the two ends of the C-shaped rings. Remember, as we talked about, we have that ciliated pseudostratified columnar epithelial tissue moving the mucus up and out, forming that mucus elevator. Remember, we talked about how in the vestibular fold, we have that sensitive mucus membrane that can cause that coughing reflex to remove dust and debris. The other place we have something like that is right down here at the branching point of the trachea. Our trachea branches to form two primary bronchi. And why do we have two primary bronchi?
Because we have two lungs.
Yeah, exactly, because we have two lungs. Excellent. Right. At this branching point is a region of the mucous membrane known as the carina. This carina has an incredibly sensitive mucous membrane to it, where if even a single dust particle comes in contact with it, it can cause a massive and very, very violent coughing reflex. If you've ever accidentally breathed in some water or something like that while you're talking, you're laughing or something like that, and you have that big, huge, massive coughing reflex, that typically occurs from that water coming in contact with this carina, causing a very, very violent protective coughing reflex to try to force that air and therefore force whatever that irritant is up and out and away from the lungs. And again, we have two primary bronchi because we have two lungs. All right. Excellent. Here we see that ciliated pseudostratified columnar epithelial tissue with some mucus glands in there. Our trachealis, as we talked about, this nice transverse section of our trachea. And we talked about why it's open in the back. And we'll look at some histology for this stuff later as well. All right. Our trachea terminates to form two primary bronchi because we have two lungs. But even though we have two lungs, the primary bronchi don't actually enter the lungs. These two primary bronchi basically lead to the two sides of the thoracic cavity, the right and left pleural cavities. But they don't actually enter into the lungs. Before they enter the lungs, they are going to branch again. Now, interestingly, these two primary bronchi are not symmetrical. The right one is typically wider, typically shorter, and typically more vertical in its orientation, whereas the left is typically longer, typically more narrow, and typically more oblique at its angle. Why might there be these asymmetries in their shapes? There you go. Due to the heart, absolutely, Izzy. As we know, the heart is not located smack dab in the center of the thoracic cavity, is slightly offset to the left side. And in fact, the left lung has a notch in it where it is offset by the heart being on that side. So we need to accommodate that by having that more oblique and longer left primary bronchi. Now, from a clinical standpoint, why might it be important to know the differences between these bronchi?
When intubating?
Absolutely, when intubating. However, in most cases, unless there's a specific lung injury, do we necessarily want to intubate all the way down to the point where we're just going to one lung versus the other?
No, but it could explain why you have unequal respiration.
Absolutely. It could affect unequal respiration. Absolutely. That could affect that way. Why else clinically might this be important? Well, let's say instead of a little bit of water that somebody inhales, what if someone inhales a chunk of food and it is blocking one of these passageways? Are both passageways equally likely to be where you'll find that congested piece of meat or pee or whatever it is?
It would probably go to the right side first.
Yeah, it's going to be much more likely to go into the right side because it's more wide. It is wider because it is more vertical. It is more likely that when a blockage occurs, it's much more likely to occur in the right side versus the left side. Excellent. Spectacular. All right. So as I mentioned, these primary bronchi do not actually enter into the lungs themselves. They enter into the left and right sides of the thoracic cavity, into the hilum region of the lungs. Right? Before we can talk more about the bronchi, we need to talk a little lung anatomy. And let's go to the whiteboard for this, because I haven't had a chance to draw yet since spring break ended. So let's go ahead and do that. Our lungs basically are, when you look at it from a superior view, have a kidney bean type shape to them, where they have a convex surface and then they have a concave surface. And those two convex surfaces point away from each other, and the two concave surfaces point towards each other. So of course, again, we have this, let's get smaller writing here to fit it all in. Convex surface, and that convex surface is going to be what is in contact with the wall of the thoracic cavity. So not surprisingly, it is referred to as the costal surface. And that is basically lateral, lateral, anterior, and posterior. Whereas the concave inner region is known as the hilum. Much like the kidneys, as we saw, the hilum is going to be where the blood vessels, the nerves, and the bronchi enter. So blood vessels, nerves, and bronchi enter into the lungs. And the specific region where that occurs is known as the root. So the root is a portion of the hilum just where the, so it's not the entire hilum, just the part where the blood vessels, nerves, and bronchi attach. Another easy way to identify the root inside of the hilum is we know something else about our lungs. We know our lungs, being contained in a cavity that is closed to the outside world, are surrounded by a serous membrane. And what do we call that serous membrane that surrounds the lungs?
Pleura.
There you go. And because it's in contact with the lungs, it is the visceral pleura. That visceral pleura, as we know, lines the outer surface of the lungs. But we know something else about our serous membranes. We know our serous membranes also are double membranes that have a parietal layer. And that parietal layer is going to line the thoracic cavity. Now I'm exaggerating the space in between them here, especially over here for my wording. But they line the thoracic cavity. And we also know that these things have to be adjacent. They're connected to each other. So what happens is our visceral pleura comes up to the blood vessels, comes up to the nerves, comes up to the bronchi, and it is there that they extend out to come and connect with the parietal. So the root is the portion that is not covered with pleura. It's where the pleura actually comes out. We've got some nice models. I've mentioned this a couple times. Let's see if it's not on the, see if I put it in the modules. If I haven't, I will definitely do that, but I'm hoping I was clever in the last class.
So the root is not covered by pleura.
Correct. Doesn't look like I have these pictures here. So I'll tell you what, I will, during the next break, I will find, unless this is it. No, this is urinary. That's not what I want. Yeah, so it looks like I don't have the pictures of the models in the class that I want. I'll do that during the next break, so I don't have to take any more time for that. I would have bet money that I had done that because those are needed. All right, well, I'll do it during the next break. Okay, so I will show those to you guys during the next break. Where were we? Whiteboard. Share. Excellent. So the root is the portion just where it connects to and comes off of the hilum. So the root is in the hilum. This whole entire concave region in the hilum, the root is just the region inside of it. All right. So we have that costal surface, we have the hilum, we have the root where the things connect. We have our visceral and our parietal pleura. If we look at the lungs from a frontal view, like we have this pretty picture from your textbook, we can see that they are cone shape. And of course, we know with a cone shape, you have an apex. It doesn't show up very well. Notice the apex basically comes up to about the level of the clavicles, where the clavicles are located up here. And we have the base, and of course, the base of our lungs sits on top of the diaphragm. So we have the apex, we have the base, we have the costal surface. Here's a pretty picture from your textbook that does a nice job of showing this, but I do want to get you a model as well. Again, notice we have this entire concave surface, which is the hilum. But within that hilum, we have this region that contains the connections for the blood vessels, the nerves, and the airways. And this specific region of the hilum is known as the root. I make a point of emphasizing this because while I really like this picture from your textbook, I find it somewhat confusing that they chose to put the tab for the hilum right here smack dab in the middle of the root. And that's not incorrect. Again, this entire concave surface is the hilum, but within that hilum is also the root. So I want to make sure we distinguish the difference between those two regions. All right. Excellent. Now, as I mentioned, we have two primary bronchi because we have two lungs. those two primary bronchi branch to form 5 secondary bronchi. Right, 3 on the right side and 2 on the left. Any idea why we have 5 secondary bronchi?
There are 3 lobes on the right and only 2 on the left.
Exactly. It's because we have 5 lobes total of our lungs. And 3 on the right and 2 on the left. And it is these secondary bronchi, 3 on the left, right, and 2 on the left, that then enter into the lobes and actually enter into the lungs. So this is where they connect to the lungs by those secondary bronchi. Notice here we can see those distinct regions, superior, middle, and inferior lobe on the right, divided by the oblique fissure and the horizontal fissure. Where on the left, because we have to accommodate the heart in that cardiac notch, we only have 2 lobes, a superior lobe and an inferior lobe, and just an just an oblique fissure on the left. There is no horizontal fissure on the left, and there's only 2 lobes on the left. All right, oops, didn't want to do that. Let's do that. What I wanted to do was erase these. So again, lungs occupy the entire thoracic cavity except the middle region. What's that middle region called again?
Mediastinum.
Mediastinum, excellent. And remember from my picture, the mediastinum does not have a serous membrane in them. Each lung is individually shrink wrapped, right? Helping to maintain the integrity and the space of this. Again, should there be damage or fluid or something like that get into this cavity, that can cause the collapsing of the lung, but you can survive that because the other lung typically will stay inflated unless the damage or the amount of blood entering in the thoracic cavity is severe. Oh, and I lost what I was writing before. All right, 3 lobes, 2 fissures in the right, 2 lobes, 1 fissure in the left, and that cardiac notch. And there we get, see, not in the picture, but in an actual cadaver. All right, we did this already. Double layer serous membrane, visceral pleura, parietal pleura, and in between, of course, is the pleural cavity. Here we see a nice transverse section. And again, if you don't like the drawing that I did, this one's much, much better, where again, we can see our visceral pleura in contact with the lungs, our parietal pleura forming the cavities lining the wall of the thoracic cavity, our mediastinum, which of course contains our pericardial cavity and our pericardium in here as well. And in the space in between, in this pleural cavity, what does it contain? What does our pleural cavity contain? Oh, there it is. Allison's got it. Sorry. Serous fluid. Excellent. Filled with serous fluid? Do we want as much serous fluid as we can get in there? Nope. We just want a thin line, thin layer of serous fluid. And don't we have a special term based on location for a thin layer of serous fluid that lines a serous membrane? Transudate. Excellent. Excellent. It is that transudate. So now the question becomes, why? What is the serous fluid doing for in here? We've talked about serous fluid before when we talked about the peritoneums of the abdominal pelvic cavity, allowing for the freedom and movement. We talked about the reduced friction and movement of the heart, of the pericardium. Why do we have serous fluid here?
A reduced friction.
Is there a lot of movement of the lungs back and forth across the surface of the ribs? There definitely is some movement. So we do want to reduce friction. But in this case, it's much, much more than that as well. Again, you're at a bar on Friday night, right? Because again, things are starting to open up and you haven't seen anybody in years. So you get into these lively debates about like who the cutest member of BTS is. And of course, we all know that it is Jimin. But if someone says something otherwise, right, that may come to blows, knife may come out, and you could get stabbed in the side. And as we talked about, this lung collapses. Why would that lung collapse? And don't pretend you don't know who Jimin is. Jimin is by far the cutest member of BTS, right? Yeah, the Jonas Brothers are back together. I mean, Jonas Brothers are okay. They're not One Direction. I still miss Zayn, but you got to change with the times. You know, Jimin, he got his degree in dance. He's very impressive. All right, excellent. Anyway, exactly, swoon. He's dreaming. Excellent. Anyway, what is that serous fluid doing in here? Why does the lung collapse when air or blood or something like that gets into this space? Anyone know?
This is pressure?
One of it is definitely pressure. One of the important things about the pleural cavity is that it has a negative pressure in it. That helps to keep the lungs inflated. But that again doesn't necessarily have to do with the serous fluid. The serous fluid, remember when we were talking about that glass of lemonade on the glass patio table? Water holds things together. And the serous fluid here in the pleural cavity produces surface tension. And that surface tension helps to keep the lung in contact with the wall of the thoracic cavity, right? Inside the airways, surface tension is bad. We don't want the two sides of our airways to connect. But outside the lung, between our lung and the thoracic cavity, their surface tension is good. So that when we use our muscles to inflate the thoracic cavity, the lungs stay stuck with it and go with it. So in this case, we absolutely positively want to have that surface tension inside. And for the record, Nick's cutest Jonas brother, Zayn, cutest One Direction, Jimin, cutest BTS. So again, that settles all of the debates when you're at the bars. All right, excellent. Okay. Did that. Come on, you guys haven't been listening. Dynamite's like every other song on the radio right now. K-pop band. You guys have to, your children aren't old enough. That's the problem, Laura. This past spring break, I spent time with Little and I've learned everything you ever wanted to know and more about BTS. There you go. Yeah, exactly. See, there you go. All right, excellent. So primary bronchi divide entering into the hilum, form our secondary, or not surprisingly, what are known as lobar bronchi, because they again go to our 5 lobes. However, the lobes aren't necessarily functional divisions. Within the lobes, there are these functional divisions known as bronchopulmonary segments. a bronchopulmonary segment is basically a unit of the lungs that is fed by one artery, fed out by one vein, and one tertiary bronchi. So this bronchopulmonary segment is a unit within the lobes that is basically fed by one tertiary bronchi, one artery, and one vein. The reason these structural units are important is not only are they functionally distinct from each other and structurally distinct from each other, but often when something like a lung cancer occurs, it occurs first within one of these bronchopulmonary segments. And if caught early enough, since there's just one artery, one vein, and one bronchi that goes into it, surgically, it's able to remove this independent unit. And if caught early enough before it spreads to other segments, it can dramatically increase the likelihood of survival from something like lung cancer if caught early enough. Now, as it turns out, there again, because of the differences in lobes and the differences in the size of the two lungs, there are 10 bronchopulmonary segments on the right side for everyone. However, for everyone, it varies somewhere between 6 and 10 bronchopulmonary segments on the left side. Some people have as few as 6. Some people have as many as 10. From a physiology standpoint, how much gas exchange can take place, how much air you can get into your lungs, there doesn't appear to be a difference that way. So it doesn't appear to have a functional difference. It's strictly a structural difference, but it's there. So again, I can tell everybody here that you have 10 bronchopulmonary segments on the right side. I don't know how many you have on the left, but I know it doesn't matter for the function. But let's do some math. We know we have 2 primary bronchi because we have 2 lungs. We have 5 secondary bronchi because we have 5 lobes. Well, guess how many tertiary bronchi we have?
16 to 20.
There you go, 16 to 20 tertiary bronchi, because we have somewhere between 16 and 20 bronchopulmonary. Notice all one word, bronchopulmonary segments. So if you'll notice, the first 3 branchings of our bronchi are not random. We have 2 primary feeding into 5 secondary, feeding into somewhere between 6 and 20 tertiary bronchi because of anatomical distinctions of the lungs. In fact, I have this great picture. Now, again, from there, it will continue to branch, and these branchings are much more random. In fact, it can undergo 21 more orders of branching on its way down to the alveoli, until we get to the smallest bronchi, known as bronchioles. And we'll talk about what defines a bronchiole in just a minute, but I want to show you this picture first. I love this picture because what they've actually did is they took a lungs, they filled it with basically gel that hardened, an epoxy basically, and it filled all of the branching, and they were actually able to colorize it. So we can see the single trachea that comes down to the 2 primary bronchi, branches to the secondary bronchi, 1 and 2, 1 and 2 and 3 over here on the right. And then you can see they actually colorized all of the tertiary and further branching for the individual bronchopulmonary segments. And so notice there are 10 of them on the right side. And notice this particular lung has 9 on the left. Now, like I said, the right side is pretty standard for everyone. 3 in the superior, 2 in the middle, 5 in the inferior. That's pretty standard for everyone. Again, how many we have on the left vary from person to person. So that's where the variation occurs. But so you don't have to memorize these particular numbers, but be able to recognize these bronchopulmonary segments. All righty. Questions on that? All right, let's talk a little bit about the histology of this bronchial tree. Here we can see this branching that is taking place at first, starting first with our primary bronchi. Here we have the primary bronchi. Notice on the left side. The primary bronchi on the left side anatomically is basically almost identical to the trachea, in that it has C-shaped cartilage rings and it is lined with a ciliated, pseudostratified, columnar epithelial tissue. So basically our primary bronchi is essentially identical, almost identical in anatomy to the trachea. But notice as we start going to the secondary and the tertiary and beyond, the anatomy changes. So what happens is the anatomy changes, the anatomy transforms. And actually, let's do this way first. And then I'll bring, oh, stop it. So notice one of the first things that should stand out to you is there's a change in the cartilage. We go from C-shaped rings to more irregular cartilage plates. Those cartilage plates are going to help to still keep the airways open. But what's the advantage of breaking up these rings into irregular shaped plates?
They can expand and change shapes in different directions.
Absolutely. Much more flexibility in being able to change the diameter, right? If you have a ring, there's only so much change in size you can have. Whereas if you have all these irregular plates, it's still going to keep it open, but it's going to allow you to expand and contrast the size. So we can get a much bigger change in the diameter of these in the smaller bronchi. Of course, that means a change in the smooth muscle. Typically, there is more smooth muscle in these layers because, again, we want to be able more change in diameter. Affecting airflow. All right. As we'll talk about, we'll talk some physics, but as we talked about before, the less air that's in contact with the sides of the lumen, the less friction you have, the easier it is for it to flow. So when you constrict it, it dramatically decreases airflow. When you dilate it, it dramatically increases airflow. So we can dramatically change the airflow as a result of it. The third change that occurs as we go down isn't something we see here in the in this particular illustration, but there is a change in the epithelium. Again, in the primary bronchi, it is a ciliated pseudostratified columnar, but as we move into the second and into the third, it changes into a simple columnar until ultimately at the end in the bronchioles, it's going to change down into a simple cuboidal. So we're going to see these changes. And remember, I mentioned the smallest of these passageways are what we call our bronchioles. Notice with the bronchioles, the way we can distinguish a bronchiole is that they have no cartilage on them, a thin layer of smooth muscle, and a simple cuboidal epithelial tissue. So again, these are the smallest, obviously the smallest of our bronchioles. And again, between the bronchioles and the tertiary, as I said, from basically here to here, we can have as many as 21 additional branches, levels of branching that can take place here as these get smaller and smaller and smaller. Kind of like a tree, and that's exactly what this is. If you were to take this bronchi and turn it upside down, you got the trachea, which is the single trunk, and then your primary branches and all the second branches that all lead out to the alveoli at the end, which I guess would be the lungs. I mean, the leaves. All right. Questions on this? All right, excellent. What's next? So there you go. We get the change in cartilage, change in epithelium, change in smooth muscle as we transition from primary all the way down to bronchioles. Our functionally distinct conducting zone ends at what we call the terminal bronchiole. Here we see an example of a terminal bronchiole right here. Notice as we look at this in the illustration, we can see that it has a thin layer of smooth muscle, no cartilage around it. And even though we can't tell, we know it has a simple cuboidal epithelial tissue on it here. These terminal bronchioles feed into a region known as a respiratory bronchiole. Notice a respiratory bronchiole is got a thin layer of smooth muscle, has no cartilage, is going to be lined with a simple cuboidal epithelial tissue. The one big difference between a respiratory bronchiole and a terminal bronchiole is notice this respiratory bronchiole has two alveoli on it. And if it has alveoli on it, it can participate in gas exchange. Now, this respiratory bronchiole with all of two alveoli on it, is this going to be the primary location where gas exchange takes place?
No.
No, it's not a trick question. Absolutely not. But if any gas exchange can take place there, then it's not a part of the conducting zone. It is instead part of the respiratory zone. So this division between the terminal bronchiole and the respiratory bronchiole is that functional division between the conducting zone, bringing the air down, moisten, filtering, and cleaning it, and where our gas exchange is going to take place. These terminal bronchioles feed into a single subunit of a bronchopulmonary segment known as a pulmonary lobule. And this is where we have our respiratory bronchioles and where we have our alveoli for our gas exchange to take place. All right. So questions on the anatomy or function of the conducting zone. All right, excellent. Then I am going to find us all the models and the pictures that we need to catch up on some of this material for the anatomy of it. And so while I find those pictures during the break, you can all do web searches for BTS, and I will meet you back here in 15 minutes. It is 10:25 right now, so we will come back, and I'm gonna have to write this down because I'm gonna have to close this window. So you're not gonna be able to see this, and I won't remember. So I'll write it down here so I will remember, but I'll write it right now so that we recall. 10:40, we will restart. All right, so at 10:40, meet back here. That's in 15 minutes, and that gives me a chance to get some of the models and pictures that we need. Any questions? All right, I'll see you guys in 15 minutes. Let's go ahead and pick up where we left off. We were working our way through the conducting zone and talking about all the anatomy and everything associated with that. So let's go down now to the respiratory zone where our gas exchange is going to take place. Again, I'm not the one here with the clicker that went through and counted them all, but they tell me that the average lung has about 300 million alveoli in them. And if you were to take those surface areas out and lay them out end to end, it would hurt a lot. It would also be about the size of a small tennis court. It is a pretty impressive surface area because, again, as we've talked about, gas exchange is passive. And since it is passive, there are things that we can do to increase the rate of diffusion. One of them is, like we talked about, increasing the temperature. Increasing that temperature helps in the gas exchange. We can dissolve the gases in water from the air. That is going to help to facilitate the gas exchange to take place. But remember, for gas exchange to take place, we want a huge surface area. And we want the gases to have to travel the shortest distance possible. So we are emphasizing these things by doing this type of stuff. And this is also why the lungs are in the inside of our body, right? Aside from the fact that you don't have a small tennis court worth of surface area on the outside of your body, this is a tiny thin membrane that would not provide a lot of protection. So all sorts of other things would get out. I would get in, water would get out, all sorts of other things as well. Unfortunately, we're not in the classroom, because if we were in the classroom, you would have the opportunity to actually feel we actually have a freeze-dried lung in the classroom, and it feels like styrofoam because most of it is air. For the size, is an incredibly buoyant and light tissue because it is mostly air. And that's what we see here when we are looking at the microscopy of it. Notice we can see some of the structures that we have already talked about. In particular, we can see a respiratory bronchiole. Notice, and let's use the thinnest line here possible, we can see a single layer of cuboidal cells that are lining this surface there as well. Well, actually not there, hold on. Those cuboidal cells that are lining the surface of this with the tiniest little bit of smooth muscle that we can see around it as well. But notice we can tell this is a respiratory bronchiole because we can also see that there is a alveolus connected to it as well. So there's that alveolus that is connected to it. So even though this is a bronchiole, it has an alveolus on it. It can participate in gas exchange. So it is a respiratory bronchiole. Notice the majority of the lungs is made up of this simple squamous epithelial tissue, forming all of the alveoli. And if you look at it like in the previous picture, it looks like a clump of grapes, right? If we cheat and go back to the previous picture, it looks like a clump of grapes, a cluster of grapes. And if you go to the store and buy a cluster of grapes, they're all held together. Are they held together by hope? What holds a cluster of grapes together?
Stem.
The stems, absolutely. Do you necessarily see the stems from the outside? No, but you know it's in there holding them together. And that is essentially what the alveolar duct is doing. The alveolar duct is basically the passageway, the hallway. The other way you can think about it, and if you don't like the stem of a grapes analogy, I always think of the hallway of a hotel, right? Basically, all that hallway is something that connects all the rooms together, right? Without the rooms, that hallway serves no function. It's just empty space. And that's exactly what an alveolar duct is. Notice as we look at the alveolar duct histologically, it basically is this big, huge, empty space, the hallway, that all the rooms, the alveoli, come off of. So notice all the alveoli come off of the alveolar duct. So the alveolar duct is the stem holding the grapes together. It is what holds the alveoli together, forming a structure we call an alveolar sac. And again, huge, huge, massive surface area to this. Here we see an illustration that kind of shows this as well. Again, notice we have our terminal bronchiole with the thin layer of smooth muscle, right? And the structure, no cartilage and no alveoli. Here we have our respiratory bronchiole, which again are bronchi, but have some alveoli on them. And they feed into our clusters of grapes, known as the alveolar sac. That alveolar sac has the stem, the alveolar duct at the center, and all the alveoli, singular being alveolus, clustered around them. And it is these alveoli where our gas exchange is going to take place. We did all of that. Excellent. If we were to look here, you see a colorized light microscopy view of this, but here we actually see a nice illustration that shows these alveoli here. We can see some of the structures and the anatomy outside of it. Alveoli are made up of a single layer, primarily I should say, of a single layer of squamous cells known as type 1 pneumocytes. So type 1 pneumocytes are simple squamous cells that form the structure, primary structure of the alveolus. And they're the ones that are beige here in our illustration. So like this one and this one and this one and here, forming the walls of it. Notice there what are going to form our respiratory membrane between the tiny little blood vessels that wrap around it and the alveolus itself. And again, notice the relative size. Remember, a capillary is so small that the blood vessels, red blood cells basically have to line up single file to get through them. So they're much, much smaller than the alveoli containing the gas that they surround. So they wrap around like fingers around a ball. Of course, if we have something named a type 1 pneumocyte, there must be a type 2 pneumocyte. In this particular illustration, the type 2 pneumocytes are these green cells embedded within the walls of our alveoli. These green cells embedded within the walls do help to form the structure, but these are called septal cells also. And our septal cells produce a substance known as surfactant. This surfactant comes down to that surface tension we were talking about. And here inside the alveolus, like inside of our balloon that we're trying to fill with air, is surface tension a good thing or a bad thing? It is a bad thing. So what that surfactant does is it helps to break up the surface tension inside of the alveolus so that the water lining the two sides don't attract each other and our alveolus doesn't collapse. Remember, we have all that moisture from the air that we've been bringing down here. It's a very moist environment in these alveoli. And so we don't want the moist surfaces of the alveoli to collapse closed and stay closed thanks to that surface tension. So the surfactant helps to break it up. So those are our two pneumocytes making up the walls of our alveolus. But notice there are also these blue cells. These blue cells are our alveolar macrophages, specialized monocytes, also known as dust cells. Being macrophages, which of course we remember are our big eaters, being dust cells by name. These are cells that migrate between the alveoli from alveoli to alveoli, and guess what their job is?
Eat pathogens?
Yeah, any harmful pathogens, any other types of irritants, dust or debris that may get here all the way to the alveoli. Even though we have all these protections, all these filters, the air that's going to get to our lungs isn't going to necessarily be perfectly clear, and especially in the forms of pathogens. Their job is to stop those before they ever get a chance to enter into our body. All right, questions on that? All right, excellent. Again, this is our respiratory zone. This is where our gas exchange is going to take place. And this is where we have that membrane that defines the inside of our body from the outside of our body. Basically, this is our air-blood barrier. Right? It's not a massive barrier, but it is still a membrane that separates us from the outside world. And as we see from the illustration, and as we've talked about before, it is comprised of our pulmonary capillary and our alveolar wall. And what type of tissue did we say primarily makes up the wall of our alveoli?
Simple squamous.
Yeah, simple squamous. And our pulmonary capillaries. And as I'm sure you remember from the first section of 431, what is the anatomy of a pulmonary capillary? What tissue forms our capillaries? Come on, it hasn't been that long. And this is a cumulative class with a cumulative final exam. What tissue makes up the walls of a pulmonary capillary?
Is it fenestrated something?
True, some will be fenestrated. Here in the lungs, when we're just dealing with gas exchange, it wouldn't be a fenestrated capillary, it'd be a continuous capillary. Yeah, it is also a simple squamous. Right? What we would call the endothelium, right? Remember that? Basically, what our respiratory membrane is two simple squamous epithelial tissues back to back. We have the type 1 pneumocytes. Of our alveolus. We have the simple squamous endothelium of our pulmonary capillary. And as we know, all epithelial tissues on their basal surfaces have a basement membrane or basal lamina. And so basically we've got that fused basal lamina of the two together. So basically we have The basal surfaces of two simple squamous epithelial tissues stuck together. Is that going to form a super massive, really thick membrane? So the part where you emphatically say no out loud.
No.
Excellent. Yes, absolutely. No, and in fact, the distance from the inside of the body to the outside of the body here in the lungs is only about 0.5 micron. Two flat cells side to side. Again, gas exchange is a vital function, and it is completely passive. We do not use any ATP in the gas exchange that takes place. So we want a surface area the size of a tennis court with the thickness of 0.5 micron to make it as easy as possible to get as much oxygen in and as much CO2 out. And it's not truly as much in and as much out. We will actually talk numbers in this case for this, but not today. All right. Questions on the anatomy of this? All right, you guys, again, like I said, it's the Monday after spring break. So again, I know, especially with an 8 o'clock in the morning class, you guys get a little bit of leeway in this, but this is a cumulative exam and there are some things we absolutely positively need to know. So let's remind ourselves of some of the things that we do indeed know. And one of the things that we do indeed know about is the pulmonary circulation. What is the job of the pulmonary circulation?
Bring oxygen poor blood to the capillaries to leave and bring in oxygen rich blood.
Excellent. So this is our, and again, we can use fancy, you're absolutely 100% correct. But remember, we also can use fancy terms now. This is our external gas exchange. Right? Absolutely. This is going to be the function of our pulmonary circulation is to get blood to the lungs for that gas exchange to take place. And conveniently enough, we learned the anatomy of the pulmonary circulation. So here, of course, is my heart. Again, I'm sure you've all missed my tremendously anatomically accurate drawings of the heart. And our job here is to get to the lungs. So let's do an incredibly realistic drawing of the lungs. There we go. Excellent. Now, remind me again what structures are involved in the pulmonary circulation. Start easy. Which side of the heart, right or left?
Left.
For the pulmonary circulation?
Right.
True. Okay, so excellent. So blood leaves which chamber, which of the 4 chambers of our heart?
The right ventricle.
Excellent. It is going to start with the right ventricle. Excellent. And what is the blood vessel that is going to receive the blood coming out of here?
Pulmonary artery.
Before the pulmonary artery. There are 2 pulmonary arteries, which indeed will receive blood, but before those 2 pulmonary arteries receive blood, what one blood vessel receives it first directly from the right ventricle?
Pulmonary trunk.
There we go. Sorry, Allison, I didn't see that you'd written it. Excellent. What is the condition of the blood as it leaves this right ventricle?
Oxygen poor.
And now we can add to this CO2 rich. Although, like I said, we'll put this in parentheses because we are going to actually quantify this with numbers. And as we all know, oxygen poor blood is blue. So we have this blue now. Blue blood coming out the pulmonary trunk, coming out the pulmonary arteries, entering into the root of my lung. And where does this pulmonary artery go?
To the pulmonary capillaries.
Right, to the pulmonary capillaries. This is going to go to, that we just talked about, our pulmonary capillary, where, of course, that gas exchange is going to take place. And let's go ahead and make that blue for consistency. In this pulmonary capillary, of course, as we've talked about, this is our external gas exchange. Which way does oxygen move again?
Into the capillaries.
And which way does CO2 move?
Out of the capillaries.
Excellent. So now at the end of this process, we have blood that is now oxygen rich and CO2 poor. That travels back to the lungs via what?
Pulmonary veins.
Excellent. And those pulmonary veins feed into what?
Left atrium.
Excellent. Oops. There we go. Hold on. Undo that. Excellent. Hopefully that wasn't too difficult. All stuff that we know and that we remember. But what is interesting is that there is actually a second circulation associated with our lungs. And this second circulation is the bronchial circulation. This bronchial circulation function is actually internal gas exchange. This is actually part of the systemic circulation. And its job is to give oxygen and nutrients to the tissues of the lungs. All right, its job is to remove wastes, just like all other systemic capillaries and systemic circulations are as well. All right. Again, notice our pulmonary circulation only travels to the respiratory membrane. Basically, our bronchial circulation job is then to go everywhere except the alveoli. Or let's be consistent, go everywhere except the respiratory membrane. the pneumocytes get all the oxygen that they need, nutrients and stuff, because again, they're after all passive, so they don't need that much ATP. They can get everything they need from the pulmonary circulation, but our bronchial circulation is going to go to the cartilage, to the smooth muscles, to the connective tissue. Of the lungs. So how would it get there? Where would this bronchial circulation begin? Well, if it's part of the systemic circulation, where would that start? Where does our systemic circulation begin? All right. What if instead I was telling you, how do you get blood to your index finger? Where does that begin? This microphone's still on,
At the
right?
heart.
Left ventricle, absolutely. This is going to start in the left ventricle. No, that's way too big. Still too big. It's going to start in the left ventricle, absolutely. And we are going to have oxygen rich and CO2 poor blood that is going to travel out of the left ventricle into what blood vessel? Although remember, we want to be more specific than just aorta. What part of the aorta?
Trunk.
What's the first part of the aorta that receives blood?
Aortic arch,
Before the arch.
ascending aorta.
There we go. Ascending aorta. Oops, there we go. Which you guys are correct, then goes into the aortic arch. Which then goes into the thoracic aorta. And off of the thoracic aorta, we then have our bronchial arteries. These bronchial arteries are then going to enter also into the hilum in the root. But they're going to, how am I going to do this? I'll cheat. I'll go past this. Here to here, out here to a systemic capillary. Oops, that doesn't need to capitalize. In this systemic capillary, we are going to have an example of internal gas exchange take place. And in this internal gas exchange, which way does oxygen go? So if I'm going to the muscle or the cartilage or the connective tissue in this lung, is it giving off oxygen or is it taking oxygen? Yeah, it's going to go into the cell. Or let's use the same term we did before. Is it going to move out of the capillary or into the capillary?
Out of the capillary.
Out of the capillary. Whereas which way is CO2 going to move?
Into the capillary.
Excellent, into the capillary. And so what that means as a result of this is we are then going to have there we go. And our oxygen poor and CO2 rich blood leaving in our, actually, hold on. I got to do this first. We're going to have our oxygen poor and CO2 rich blood in these, put that there, in our bronchial veins coming out of that systemic capillary. And where are those bronchial veins going to go? Well, where does
Vena
oxygen?
cava.
Right, you'd want it to get back into the superior vena cava, right? Because you want to get it back to the right side of the heart, because that's where oxygen poor, CO2 rich blood goes, right? So you'd want to complete the cycle of our systemic by doing that way. But it turns out bronchial veins are lazy. Yeah, they could carry this blood back to the heart, but you know what? Those pulmonary veins are already going back there. So instead, what our bronchial veins say, hey, pulmonary veins, do me a favor, take this blood back to the heart. So these bronchial veins actually feed into the pulmonary veins. And our pulmonary veins then carry all that blood back to the left atrium. So notice with this system, there are two circulations that feed into the lungs, but only one circulation that comes back from the lungs. All right. Notice one more minor implication to this. Hold on, I want that to stay big. Not that.
Does this mean it's putting the oxygen poor blood into the pulmonary vein, which should be rich?
Exactly. So that was the last point that I'm going to make, and I'm glad you made it for me. Absolutely. What this means is that the blood that returns to the lungs, to the left atrium, has slightly less oxygen in it than when the gas exchange first takes place. And we will actually put numbers to this on, I think on Wednesday, so we can actually see the actual implications of that. But you're absolutely correct. There is going to be, the blood that comes back to the heart is slightly less oxygenated than it was when the gas exchange takes place, because that oxygen poor blood is being dumped into it. It's not a ton, but it's enough to drop it a little bit. A little less than 4% drop in the oxygen carrying capacity of the blood as a result of that. All right, so we did it with the pretty pictures. Let's do it with the words. Two circulations, pulmonary and bronchial. Pulmonary arteries, we talked about the condition of the blood and feed the pulmonary capillaries. And our pulmonary veins come back. We talked about the condition of that blood. So again, oxygen poor blood, CO2 rich blood goes to the lungs. Our gas exchange takes place, and an oxygen rich, CO2 poor blood comes back. But we also have these bronchial blood supplies. The bronchial arteries, basically systemic, come off the thoracic aorta, carry oxygen-rich lung blood to pretty much everything in the lungs except the alveoli, where systemic gas exchange takes place. and the bronchial veins then anastomose with the pulmonary veins carrying them back. So pulmonary veins carry all the blood back. Now we have, and this was one of the things that I was looking for. So let's do this first. A great model. It's one of my favorite models in the classroom that does a really, really excellent job, almost perfect, except for one hugely, massively wrong thing that it does. Showing us one of these individual segmented lobules inside of a lung. So here we have our terminal bronchiole coming down. Let's make this bigger. Terminal bronchiole coming down, feeding into a respiratory bronchiole, which has some alveoli on it, into an individual lobule, where we can see the connective tissue that is separating it and compartmental in it. And then here we see some of the alveolar sacs. And we see the blood vessels feeding into and out of this. On the outside, it's rounded like this. On the inside, or the other side of it, it's flat. So here's a close-up view where, again, we can see our terminal bronchiole feeding into a respiratory bronchiole. And let's look here at our blood vessels. Here we have a dark-colored blood vessel that is coming down, follows the alveolar ducts and feeds into the alveoli. What blood vessel do you think this represents? Well, it's going to the alveoli and it's blue in color. So what might that indicate to you? What is the condition of the blood if the blood vessel is bluish colored?
Pulmonary artery.
Yeah, this is the pulmonary artery. Feeding into our pulmonary capillary. And what type of gas exchange takes place in this pulmonary capillary?
Oxygen moves into the capillary and CO2 moves out.
Excellent. And what do we call that type of gas exchange?
External.
There
Yes.
you go. External gas exchange, where oxygen moves. And let's change the vocabulary just because it amuses me to do so. Out of the air into the blood. Whereas CO2 moves out of the blood into the air. Excellent. Again, as long as you are describing what structure, what medium it's going into or out of, capillary, alveolus, blood, air, again, there's lots of ways to say it correctly. Just don't say in or out. You need to tell me what it's moving into or what it's moving out of. Excellent. Notice then it becomes oxygen rich, CO2 poor, and then leaves via what blood vessel right there?
Pulmonary vein.
Pulmonary vein. Excellent. All right. That is great. That is perfect. That is fine, except for one major detail. I didn't mention it back here in the picture, but let's run back to the picture real quick, although I'm going to lose this if I do that. So let's, we can take an image of this, save that. Actually, we can go ahead and do that here. The only problem with this model is that our pulmonary artery follows the bronchi all the way down, whereas the pulmonary vein branches off from it. So notice they have that reversed here. So really, this blood vessel should be draped over the top of the bronchi, and this blood vessel should be offset to the side. So the top of this is wrong in the orientation of the blood vessels, but everything else here it shows is perfectly fine. All right, so that is the one major error in this model. But as long as you ignore where the two blood vessels is, everything else on this is perfect. All right, but I want you to notice one other thing. Notice there is a second red blood vessel that is kind of a different color. Notice this red blood vessel doesn't go to the alveoli. It stays in the connective tissue septum. And in fact, if we follow it down to the bottom of the model, we see that it feeds into a capillary in the connective tissue away from the alveoli. Guess what blood vessel this is?
Bronchial vein.
Excellent. And notice that bronchial vein feeds into what type of capillary?
Systemic.
And in this systemic capillary, which way does oxygen flow and which way does CO2 flow?
Oxygen out and CO2 in.
Out and in of what? What is CO2 oxygen flowing out of?
The capillary.
Okay, excellent. Out of the capillary, and CO2 is going into the capillary. And let's finish it out. Out of the capillary into the tissue. And this is out of the tissue and into the capillary. Structure to structure for that there. Excellent. Notice it then feeds out, in this case, into a very small, running out of room here, we'll write it over here. Very small blood vessel. What would the small blood vessel that the capillary feeds out into be? Oh, wait, hold on. This wasn't the bronchial vein. Did you guys say it right? Did I write it wrong?
Yeah, that's the bronchial artery, right?
Yeah, that's a bronchial artery. Right, because the bronchial artery away from the heart, bringing, again, systemic, so it's coming to a systemic capillary, and then it feeds out into the bronchial vein. However, notice the bronchial vein, anastomosis, feeds into our pulmonary vein. And remember, the pulmonary vein is what then carries the blood back to the heart. Right? It's like Thunderdome. 2 blood vessels enter, but only 1 leaves. And again, I know most of you don't get that reference, and I don't care. All right, excellent. So great model, does a great job of showing, except for that one major problem up at the top, which if you ignore, it's a great model that shows these 2 circulations really, really nicely. All right. While we're here, the other pictures I wanted to show you. Again, I just tried to find some quick models that do a nice job of showing this area epiglottic fold. This isn't the most detailed model, but notice even with this not most detailed model, we can still see the 4 bumps of the cuneiform and the corniculate cartilages, the aryepiglottic fold coming down from the epiglottis. Although I think this next one's a little nicer. Oh no, actually this is the one I wanted. Yeah. So you notice this one, you can even see the arytenoid cartilage, its corniculate forming that inferior bump. And then right here, we see that second bump, which is the corniculate. And then again, this one's not as detailed, but again, we can see this is the corniculate where it would be sitting on top of the arytenoid cartilage. Here we see the second bump of that cuneiform. Here we're seeing the epiglottis, the epiglottic fold. And notice this one does a nice job of showing us the vestibular fold and our vocal fold on this as well. We can see the hyoid bone on the top, thyroid cartilage, cricoid cartilage sitting on top of our trachea. But notice it shouldn't be cartilage back here on the back of our trachea, because remember, our C-shaped rings don't go around. So again, not a perfect model, but it's the best I could do in 15 minutes. I was more interested in finding these pictures because I wanted to show you that. And so then I just threw these on as an extra. But again, you can do a Google search and show these really nicely. This is actually a picture from the classroom of a model that we have in there that I like a lot. But there's some other really good ones that both have the mucous membrane. And if you just want the cartilage, there are plenty of mixed pictures and models that show that as well. All righty. Questions on any of that? All right, I believe, yep, that is everything that I wanted to cover today. We've gotten the anatomy taken care of. We have talked about the blood supply. And so starting on, you guys seen this or am I just seeing this? There we go, make sure I'm sharing this. Excellent. So again, we've done all of the circulations. We've done all of the anatomy. We've got all the anatomy out of the way. So we can start talking about some of the functions of these things, starting first with pulmonary ventilation, the muscles and the activities involved with that. We're going to start getting into some physics, because while, as we all know, chemistry is an incredibly stupid and pointless study, physics is beautiful and elegant and important. And we will spend some time talking about that. And seeing how this whole thing works. Because as we know, pressure makes the world go round. So we'll learn some of the laws associated with that. All right. Questions on any of that? All right. That is actually all I have for you guys today. So thank you very much. Welcome back from break. Remember, I will have office hours. I will take a quick break to have some lunch, but then I'll be back. I will be releasing the exams. So if you have any questions, starting at noon, I'll start my office hours early if anybody has any questions and wants to come and meet and talk about it. So I'll be releasing the exams just now. So you guys have a good day, and I will either see you in my office hours or see you on Wednesday. All right, take care and bye.
Thanks.